some idea of when you will get the results from your exam from last week. So hopefully by the end of next week, I should have the comments and the correction from all the teachers. So if I have them on Thursday, sorry, then I will give you the results on Thursday. And in that case, probably, because I cannot put the results, I think, directly on the internet, which is accessible to everybody, so it has to be here. So I will probably come with a list and with the results rather than putting it on the web. So that's if, yes?
You're going to put the results with our
IDs.
Yeah, IDs without our names.
Yeah, but there was some, we did that last year,
Yeah.
but there were some issues with that as well. So anyway, I think if I have all the corrections for next Thursday, I guess some of you may want to see why they get a particular score. So what I can do is come a half an hour earlier before the lecture, and then if you want to see your exam, you will be able to see your exam. I need to keep the original though, but you'll have the score and the correction at the same time. If it's not next week, it will be the week after. Okay? So today I'm happy to welcome Stéphane Lefrançois. He's a researcher at the Institut Armand-Frappier in Montreal. His research is aimed at understanding mechanisms regulating intracellular sorting and trafficking, mainly in the context of neurodegenerative diseases. And I think he's particularly well suited for the title of the lecture today, which is Reticular Trafficking. So thank you for coming and enjoy your lecture.
Okay, so today we're going to talk about vesicular trafficking. As you can tell by my name, I'm Francophone. I was asked to give this in English due to the composition of the class, but feel free to ask any questions or make any comments in French. That's no problem. Okay? We can keep this rather informal. If you have questions or concerns or comments, or you're completely confused at one point, please interrupt me and we can discuss any issues you may have. All right? Okay, so what is vesicular trafficking? So, on a little side note, the field was quite excited in 2013 with the Nobel announcements. Does anybody know why? Because vesicular trafficking won the Nobel Prize in Medicine. So James Rothman, Randy Schekman, and Tom Sudoff sort of were the pioneers in elucidating all these trafficking mechanisms using yeast and using mammalian cells. And bottom line, vesicular trafficking is the movement of cargo through the cell with the formation of trafficking vesicles. So if you look here at the ER, there's a trafficking pathway between the ER and the Golgi. It's mediated by this COP complex. So leaving the ER, you have COP2 that forms these vesicles. These vesicles will travel to the ergic. They'll eventually fuse here in the Golgi, and they'll deposit protein content into the Golgi. Now, proteins that are necessary to maintain the functioning of the ER sometimes escape, and they need to be brought back. And they'll use the COP1 system, where again you'll have these vesicles that will bud from the Golgi, travel back to the ER to return enzymes and resident proteins in the ER. Okay? So this is all you're going to hear about COP trafficking today, because all of my work deals with these post-Golgi sorting events. So so we'll focus on these post-Golgi sort of traffic. Okay, so if you have this COP system, COP1 and COP2, between the ER and the Golgi, post-Golgi trafficking depends on another coat protein called clathrin. Okay? So today we're going to talk a bit about clathrin. We're going to talk about how these clathrin-coated trafficking vesicles are made and how the cell regulates their formation in quite a bit of detail. Okay? So bottom line is you can have clathrin that'll bud from the TGN, so this later Golgi. You can have vesicles that'll traffic either to the cell surface, to different endosomal compartments. From the endosome, you'll have clathrin-coated buds that can traffic from the endosome to the lysosome, back to the cell surface, or back to the Golgi itself. Okay? So this is what clathrin looks like under an electron microscope. It's a triskelion shape, and it forms these little basket-type shapes, and it can deform membranes and form these vesicles. Okay, so here's one clathrin triskelion, and they sort of self-assemble into these vesicular shape. Okay, and if you look at sort of the lifespan of these clathrin-coated vesicles, you have a donor membrane, which in this case could be, say, the Golgi. You have protein that needs to be sorted out of the Golgi and trafficked to this acceptor membrane. So it could be the endosome, it could be the plasma membrane, it could be another cellular destination. Okay? So the cargo will bind a transmembrane receptor. The transmembrane receptor will start binding to a variety of proteins that are found in the cytosol, and eventually clathrin. You'll have the formation of a bud. The membrane here will be cut. You'll have a vesicle with the cargo protein. So the whole point of this system is to move this purple dot from point A to point B. Okay? At a certain step, the vesicle will uncoat, so the coat proteins in the clathrin will fall off. The vesicle will start moving towards its destination. And then there's this machinery at the acceptor membrane via these snares, which we'll talk a little bit about, that the vesicle will bind to the acceptor membrane, the vesicle will fuse, and the cargo will be released into the acceptor compartment. Okay? So a little bit more close up. Here's the receptor. The red dot is the cargo that needs to be moved. Here are the adapter proteins, and here's clathrin. You can see that at a bud, there's an enrichment of the cargo with the receptors, and the clathrin can then start deforming the membrane to start creating a vesicle. Once the vesicle is almost formed, you have this neck region here that needs to be cut, and a protein called dynamin will come in and cut the membrane so that the vesicle can then bud off. And here you have an electron micrograph where a mutant form of dynamin is expressed in this cell, and you can see the vesicles are trying to bud, and the dynamin is being recruited, trying to cut the neck here, but because it's non-functioning, it can't, so you have this accumulation of these budding vesicles. So how does dynamin work? There's two proposed models, and we don't really know which one's correct, but if you think of my head as the vesicle that needs to depart, so my shoulders here are going to be the donor membrane, dynamin is going to wrap around my neck, and it'll either squeeze until the membrane sort of pinches off and it separates, or dynamin will just create a force that pushes my head up towards the ceiling until the membrane thins out and the vesicle just pops up. Okay, so those are the two proposed models, and nobody really knows which one of the two is correct right now. Okay, so once dynamin's cut, you have the free vesicle. The vesicle will uncoat. So here you have the vesicle with the receptor and the cargo protein. And then the docking is going to be determined by a group of proteins called snares. So within the trafficking vesicle, so here's the cargo and the receptor with the vesicle membrane, you have a protein called a V snare, so a vesicular snare, which is going to look for tethering factors and a matching snare on the target or acceptor membrane. When the V snare and the T snare, so target membrane snare, come together, it forms like a zipper. So the snares sort of collapse onto one another, and they bring the vesicle in towards the acceptor membrane. and it results in fusion of the vesicle with the target membrane. Okay, and the cargo can be released, and the receptor is there, and the two snares are zipped up together. Now, the snares also play an important role in determining where the vesicle goes. So there's matching pairs of V snares and T snares on different membrane compartments. So for example, if you have this red sort of jagged snare versus this red snare that looks like a little C here, the vesicles will go to the target membrane and they'll only bind with the corresponding T snares so that it determines specificity. So this vesicle will never go to compartment A, while this vesicle will never fuse with compartment B. So for example, this vesicle might be leaving the Golgi and going to the endosome. So these two snares. This one might be going to the plasma membrane, and you have these two snares. So cargo destined for the plasma membrane will never fuse with the endosome. Okay? So far, so good? Yep. Oui. So they sort of stay together and then they'll be separated, and there's a machinery that will retain the T snare within this membrane, and the V snare gets recycled back to the donor membrane. Okay. So now, as promised, we're going to focus on this post-Golgi sorting. So you have the Golgi, and the area of study in my lab is really these trafficking pathways, so Golgi to endosome and endosome back to the Golgi, and how these clathrin adapters and clathrin participate in moving cargo back and forth. So, clathrin is sort of the main player in the vesicle, in the formation of the vesicle, but the cell wants to regulate packaging cargo and receptors into specific clathrin-coated vesicles. And the way they do that is with these cytosolic sorting proteins. So these are all clathrin adapters. So they, at one end, will bind a protein that needs to be trafficked, and at the other end, they'll bind clathrin and other accessory proteins responsible for making the vesicle. So they bridge cargo with clathrin. Okay? So there's three main types. There's these multimeric adapter proteins, which are composed of multiple protein subunits. There's the monomeric adapters, which is just one protein with different domains. And there's retromer, which is a pentameric complex, which is involved in endosome to Golgi trafficking. Okay? So now, if you have cargo here, so this is the red dot, these are proteins that need to go to the lysosome. So they're proteins that function at the level of the endosome, so they need to get to the lysosome. However, they can't bind the cytosolic protein, so they can't bind these adapters, they can't bind clathrin themselves, so they'll bind a receptor. Okay? You have the cargo binding the receptor then can bind these clathrin adapters. You can make clathrin-coated vesicles. The vesicles will butt off, they'll traffic to the endosome, and the cargo can then move on to live in the lysosome. Okay? So this step is primarily mediated by these monomeric adapter proteins. So they're the GIGA. So they're the Golgi localized gamma adaptin ear ARF1 binding proteins. Okay? We'll just call them GIGAs because I have a hard time remembering them. And there's three forms. So there's GIGA 1, GIGA 2, and GIGA 3. And from what we know so far, they all seem to do the same job. So if you knock out one, the others will sort of compensate. You need to knock out all three if you really want to block trafficking. Okay? So they're composed of a VHS domain, a gap domain, a hinge domain, and a guide domain. Now, what are these cargos? The little red dots that I keep telling you about. So these are all these proteins that are going to live and function at the level of the lysosome. So you have cathepsins, you have various flavors of cathepsins, different proteins that are involved in degrading proteins, lipids, DNA, all kinds of other things that get targeted to the lysosome for degradation. Okay? So these guys have got to move from the Golgi to the lysosome. Okay. So what's the green box? So there's 3 receptors. We'll divide them into 2 groups. So there's the mannose-6-phosphate receptor. The CI and the CD are not particularly important in this case. So at the Golgi, they're known to bind various flavors of cathepsins, other proteins such as PPT1, and they also play a smaller but significant role at the cell surface where they can bind and internalize proteins such as renin and IGF and lead to their degradation at the level of lysosomes. Okay, so one receptor is the mannose-6-phosphate, then you have sortilin, which is another receptor, and it'll bind proteins like rosapsin and GM2 activator proteins, and at the cell surface as well, it'll bind neurotensin and lead to its degradation. Okay, so mannose-6-phosphate receptor, sortilin, so they're type 1 transmembrane domain proteins. So if you have the membrane here, transmembrane domain, this is inside the vesicle or the lumen of the organelle where there's the cargo binding domain. So these proteins will bind here. And then you have the cytosolic tail, which is able to bind to the different adapter proteins and link sortilin or the mannose-6-phosphate receptor then to clathrin to form trafficking vesicles. Okay? And there's 2 prominent signals in the tails of these receptors. So there's this tyrosine motif, so named because of the tyrosine and the rest of these amino acids, and there's a dileucine motif, which we'll discuss in a moment in their role. So here's the tail of sortilin, the tail of the mannose-6-phosphate receptor. They both have this tyrosine motif and they both have this dileucine motif. From the point of view of the soluble cargos like cathepsins, it's the dileucine motif that's important in their trafficking. So what does this dileucine motif do? Well, it'll bind to the VHS domain of the gigas. Okay? So you have the receptor binding cargo, here's the tail in the cytosol, here's the dileucine motif, it'll bind the VHS domain of the gigas. And that's the signal, so think of it like an address, or you're addressing an envelope and you're sending the envelope from point A to point B. So the dileucine motif, through its interaction with the VHS domain, is going to send the cargo protein from the Golgi to the endosome. Okay? So how do we know that the VHS domain binds these signals? Have any of you ever done a yeast-2 hybrid experiment in the lab? Anybody know what a yeast-2 hybrid experiment is in the lab?
So we're detecting interaction between proteins.
Right. So what's one main advantage to yeast-2 hybrid compared to, say, another protein-protein interaction method? So how else can we test protein-protein interactions?
BIFC. BIFC.
BIFC?
Bimolecular fluorescence complementation.
Ooh, yeah, that's
FRET.
That's heavy duty, man. Yes, okay. I was thinking more co-IP, but yeah, okay, that's a good technique too. So let's compare yeast-2 hybrid to co-IP instead of, so I'll show you some FRET results later. So what's an advantage to yeast-2 hybrid compared to co-IP?
Direct interaction.
Direct interaction. So yeast-2 hybrid, you know that if you're having an interaction, it's the 2 proteins binding directly. Co-IP could be mediated by some other protein that you're not aware of. Okay? So the way the assay works is you put in 2 proteins into the yeast, you ask the yeast to express them, and then the yeast can grow on selected media if there is an interaction between the 2 proteins of interest. Okay? So if you have the tail of the mannose-6-phosphate receptor, here are these 2 dileucine motifs and the VHS domains of giga1, giga2, and giga3, you see that they're growing. If you mutate the 2 downstream residues to alanines, you maintain the interaction because the interaction is mediated by these 2 dileucines. If you mutate the dileucines or you get rid of the 2 dileucines, you lose the interaction. Okay? And the same thing was done with sortilin. So if you delete the 2 dileucines, you lose the interaction. Okay? So dileucine motif, VHS domain of nucleus. Okay, so here's the membrane, here's our receptor, cargo binding to the receptor, and the cytosolic component that's going to bind clathrin gets recruited to the membrane and binds a dileucine motif in the receptor, and you can start forming this clathrin-coated bud. Okay? Now, the cell doesn't necessarily want this interaction to go on all the time. So you want to be able to regulate that interaction. And you want to be able to regulate when gigas are going to bind the membrane and bind the receptor. All right, so how are we going to do that? So that's through ARFs. Okay, so these are the cytosolic G proteins that get activated from a GDP to a GTP. They'll bind the GAP domain. They'll glue the gigas onto the membrane, and the VHS domain will then be able to interact with the tail of the receptor via the dileucine motif. Okay? So here's again some yeast-2 hybrid experiments showing that the VHS GAP domain can bind to ARF1. And these pulled-down experiments show the same thing. So the ARF1 binding site is on the GAP domain of the gigas. So VHS domain will bind the dileucine motif. The GAP domain will bind ARF1. And just to show you that you really need ARF1 to recruit the gigas, so here's an immunofluorescence experiment. There's a cell here, and there's a cell here, and they're stained for giga3 in this case, and you can see nice giga3 staining on the Golgi of these cells. If you add VFA, so VFA is a fungal metabolite that will block the conversion of ARF1 GDP to GTP, so you have no active ARF. You see that the gigas aren't recruited to the membrane anymore. And if you wash out the VFA and you allow a bit of time to go back, you can see that giga3 can be recruited to the Golgi of these cells. Okay? This is another example. This is ARF1. It's a dominant negative mutant that's never activated. And if you overexpress this mutant in cells, you can see that giga3 isn't recruited in this cell compared to this cell, where giga3 is, and there's no ARF1 here. And giga recruitment is specific to ARF1 and not other ARF family members such as ARF6. If you overexpress this ARF6 mutant, giga3 is still nicely recruited to Golgi membranes in these two cells overexpressing ARF6. Okay? Any questions? Okay. So now we've initiated a trafficking vesicle that's budding from the Golgi. So the cargo is binding the receptor. The receptor is the transmembrane domain protein. What motif is binding what in the gigas? Dileucine motif in the receptor is binding the VHS domain of the gigas. ARF1's been activated from GDP to GTP, and now you're recruiting gigas, you're enabling the binding. The gigas are going to bind clathrin, and they form the trafficking vesicle. Okay? Now the receptor cargo complex is moving towards the endosome. Okay? So now, here at the level of the endosome, the cargo is released from the receptor. The cargo is going to move on to the lysosome. The cell doesn't want to waste this receptor, so it wants to bring the receptor back to the Golgi so that it can bind more cargo, bring more cargo here, and eventually be delivered to the lysosome.
What was ARF1 purpose exactly? Like, is it localizing it to the membrane, or
Yep.
how do you activate exactly?
Pardon me?
When you see it active, when it's active and then
Yep.
it recruits it, is that all it's doing is bringing it there so it can perform its function?
Right, so the gigas, there's different models, but the gigas could be just sort of coming on and off the membrane, and only when you have active ARF1 will the giga be stabilized onto the membrane to then interact with the receptor. The other,
Is ARF1 binding a transmembrane protein to keep it there?
no, so ARF actually has this myristylated residue at the N-terminus, which the cartoon doesn't do it justice, but this myristyl group, which is a lipid modification, is hidden within the core of ARF when it's inactivated. When it gets activated, the myristyl group is open and it can plug itself into the membrane via that lipid modification.
For
for the previous slide, or no, the slide with where you see the retrograde and retrograde. Yeah, the EP1. I don't remember what means EP1, but
We didn't discuss it.
okay.
Yeah, probably should remove it. So, so one of the criticisms, and I know your supervisor always thinks like, wow, protein trafficking is really difficult because there's a lot of proteins and a lot of protein-protein interactions. So although I'm telling you the gigas are this pathway, which is true, AP1 also plays a role, but I'm just simplifying it. So as far as you guys are concerned, gigas, Golgi, the endosome. Okay, so this dissociation here, can anybody tell me why the cargo comes off the membrane at the level of the endosome, but it can bind the receptor here at the level of the Golgi? pH, exactly. Okay, so as we progress through the endomembrane system of the cells, you have a very sort of neutral pH at the level of the ER, and it starts getting a bit more and more and more acidic as you go through the Golgi, and by the time you get to the early endosome, it's getting more and more acidic, and as it's moving towards the lysosome, it gets very acidic. So sortilin, for example, can bind this cargo at this more neutral tending pH, but it can't here at this more acidic pH. Okay? And as a bonus question, why is the lysosome so acidic? What's the purpose of the lysosome being so acidified?
Destroy it. To destroy any proteins, anything that goes in.
Right? So, but why?
There is a pump, H plus pump.
Yeah.
Yep, exactly. So there's a proton pump that's acidifying this organelle. So the enzymes like catepsins, for example, work best at this highly acidic pH for two reasons. One, as they're moving through this membrane trafficking, you don't want catepsins chewing up anything while you're in the Golgi. So they only really function at this more acidic pH. And two, if ever the lysosome ruptures and the content spills out into the cytosol is very basic. So those proteins won't be affected, so the cells won't get sort of endo-digested. Okay, so here we have sortilin again, retromer. So the dileucine motif is going to bind the VHS domain of the gigas, and the tyrosine motif is going to provide the address and the trafficking mechanism the other way. So if dileucine is working Golgi to endosome, in this case, tyrosine motif is working endosome back to the Golgi, and it's binding retromer. So if you look here, sortilin's being expressed. If you do a co-IP experiment here with sortilin MIC and you stain for BPS26, so one of these retromer subunits, you see that the wild type receptor is binding retromer, but the tyrosine motif here, if you change this tyrosine to an alanine or the leucine to an alanine, you block the interaction with retromer. Okay? So tyrosine motif is going to bind retromer, and it's going to mediate the trafficking of the receptor endosome back to Golgi. Okay, so now we're at the level of the endosome. Here's the receptor. The cargoes come off. Retromer is going to be recruited. It's going to bind the tyrosine motif in the tail of the receptor, and the receptor can leave the endosome and go back to the Golgi for another round of trafficking. So now, if gigas were the recruitment of the gigas were regulated by a small G protein here, there must be a small G protein regulating retromer recruitment. But what do you think happens in the cell if you block retromer function? So retromer is taking the receptor, it's binding it at the level of the endosome, and it's bringing it back to the Golgi. So if you deplete retromer from the cells, what's going to happen to the receptor? It gets degraded. Okay, so if you block retromer, the receptor starts to accumulate, it's got nowhere to go, and eventually it goes off towards the lysosome. And if you deplete any of the retromer subunits, so if you do siRNA against UPS26, and you do a pulse cyclohexamide chase assay, you see that the receptor over a 6-hour period is very stable. But if you get rid of retromer, all the way at time point 0, you have much less mannose-6-phosphate receptor, and by about 6 hours, you completely lose the receptor from the cell. Okay? So without retromer, the receptor is not recycling, and it gets degraded. And by immunofluorescence, if you deplete SNCX1 and SNCX2 by siRNA, so these cells up here have been depleted, these ones have not been depleted, and you look at the staining of the mannose-6-phosphate receptor, there's very little mannose-6-phosphate receptor in these cells, while these cells have a fair amount of the receptor still intact. Okay? So, we want to control this recruitment, so we don't want retromer just constantly binding the membrane and binding the tail of the receptor. So there's another small G protein. So for gigas, it was ARF1 in the tail of the receptor. Leucine, tyrosine. To control that interaction and to control trafficking, you have a small G protein. At the level of the Golgi, it's ARF1. At the level of the endosome, it's Rab7. Okay? So, just to show you that Rab7 is actually involved in recruitment of retromer. So if you have control cells or Rab7 depleted cells, and you look at VPS26 staining, you see this punctate endosomal staining, and if you deplete Rab7, you lose this endosomal recruitment of retromer, as shown here by this VPS26 staining. So AP3, we haven't really talked about it yet, coming up. So it's an adapter protein at the level of the Golgi, and maybe a little bit endosome. It's not affected by Rab7 depletion, and neither is AP1. Okay? So Rab7 specific for the recruitment of retromer. Okay. So now the last group of proteins, the multimeric adapters. So there's AP1, 2, 3, and 4. And they're all these multimeric adapter protein complexes. So they all have a large beta subunit. So beta 1, 2, 3, 4. They have a medium subunit called mu, 1, 2, 3, 4. And a small subunit called sigma, 1, 2, 3, 4. And then they become a bit more divergent with this other large subunit where you have gamma for AP1, alpha for AP2, delta for AP3, and epsilon for AP4. Okay? So, where do they work? So, AP1, let's sort of disregard it for this context, but it is involved in endosome to Golgi trafficking. AP4 is going to sort proteins from the Golgi to the basolateral membrane of polarized cells. AP2 is going to be involved in endocytosis from the plasma membrane to the endosome. And AP3 functions in this pathway as well for another class of proteins which we're about to discuss. Okay? So just to sort of give you an idea, but I don't want you to focus on all of these adapters. Just keep in mind the gigas, retromer, and AP3 now that we're about to discuss, okay? So what does AP3 do? So the protein content at the level of the lysosome has these soluble proteins like cathepsins that are involved in catabolism. But the lysosome also has integral membrane proteins. So for example, LAMPs, which are highly glycosylated proteins that are transmembrane proteins, that the majority of the protein is found in the lumen of the organelle, and they're highly glycosylated to form a special layer protecting the lysosome from the degradative enzymes inside. So the LAMPs sort of serve this function as a barrier preventing the cathepsins from chewing through the lysosomal membrane to escape outside the lysosome. Okay? So you have a bunch of other of these transmembrane domain proteins. So if it's a transmembrane domain protein, it doesn't need to bind a receptor to traffic. It's got its own machinery pre-built. Okay? So you have the bulk of LAMP2 within the lumen of the membrane. You have a transmembrane domain, and you have the carboxy terminal end of the protein, which is in the cytosol. Okay? And they have a couple of different signals. So they also have tyrosine signals, and they have dileucine motifs as well, okay? So different proteins have different types of trafficking motifs. In this case, because the cell wanted to confuse us all, both of these motifs serve as Golgi to endosome trafficking. Okay? It's not clear why tyrosine motifs for the sorting receptors work in the other pathway, but in this case here, we can focus on these dileucine motifs. So the multimeric adapters can bind both these dileucine motifs and these tyrosine motifs. Okay? And the tyrosine signals on, for example, AP3 will bind the mu subunit, and the dileucine signals will bind a pocket formed by delta and sigma 3. Okay? And if we go to our yeast 2 hybrid experiments, you can see here that these proteins that contain a dileucine motif, so LIMP2, you need to express gamma and sigma for them to bind, while as LAMP2, which has a tyrosine motif, will bind straight to mu3. Okay? And this is shown here on this yeast 2 hybrid. Okay? So now, you have the membrane of the Golgi here, you have LAMP that needs to go to the lysosome, you have AP3 that's going to be recruited to the membrane, bind the motifs here, so this dileucine motif, and it'll bind clathrin. So what happens if you don't have AP3 in the cell? So this is another one of these proteins that gets trafficked through AP3. So it goes to the lysosome, and if you look at the cell surface, there's very little of this CD63 on the cell surface. If you have AP3 deficient fibroblasts, CD63 doesn't get targeted directly from the Golgi to the endosome. Instead, it sort of wanders around the cell a bit aimlessly, and it ends up a lot on the cell surface. So if you do FACS analysis, you can see that in AP3 deficient cells, CD63, LAMP1, and LAMP2, so all these proteins are trafficked by AP3. In the deficient cells, you have a lot of it on the cell surface, while as the mannose-6-phosphate receptor, which uses what to traffic? So how does mannose-6-phosphate or sortilin get from the... Yeah, but yes, but that's from endosome to Golgi, from Golgi to endosome. Yeah, but from the Golgi to the endosome, the receptors will bind the gigas to traffic. Okay, so you see that these guys don't end up on the cell surface when you lose AP3. Okay, so if AP3 is coming onto the membrane, you obviously have a small ARF, a small G protein that's regulating this interaction at the level of the Golgi. And which small G protein is regulating recruitment at the level of the Golgi? ARF1? ARF1. Okay, you can see that AP3 can interact with ARF1. So if you do a co-IP here or a yeast 2 hybrid, you see that AP3, both through this pull-down assay or through this yeast 2 hybrid, can bind activated ARF1 very well, and that the binding again is through this sigma delta binding pocket. Okay, and again, here if we treat the cells with DFA, so this is this fungal metabolite that will block ARF1 activation. You can see AP3 here staining nicely, level of the Golgi and in some puncta, AP1 as well. And if you add cells to BFA, you see that AP3 and AP1 end up in the cytosol, so they're not being recruited. So you lose the ability to recruit when you don't have active ARF1. Okay, so now, you should all be hopefully asking the question, so at the Golgi, you have ARF1, an activated ARF1, that's recruiting AP3 and gigas. So now, if you're activating ARF1, and let's say you want to move LAMP from the Golgi to the endosome, you're going to need to recruit AP3. But you don't necessarily want to recruit gigas, because you're moving LAMPs. But you're activating ARF1. So if you're activating ARF1 at the Golgi, shouldn't you be recruiting the gigas? So how can the cell regulate the activity of ARF1 to make sure you're only recruiting AP3 or gigas at the level of the Golgi? Okay? And that's through a group of proteins called the GEFs. Okay, so ARF1 in its inactive GDP form will be activated to GTP. Okay? This will allow it to bind the membrane and to recruit either AP3 or the gigas. And the activity or activating ARF1 from GDP to GTP is done by a GEF, a guanine nucleotide exchange factor. Okay, and at the level of the Golgi, there's two GEFs. There's big 1 and its homolog big 2, and there's GDF1. Now, you have this active ARF. You don't want ARF to be active all the time. So I'm sure some of you work on cancers, and if you have these proteins that get turned on, and it's always on, what happens? You get cell growth, weird migration, all kinds of different things, so that's not good for the cells. So you want to shut off ARF1, and that's done through a GAP, a GTPase activating protein, and for example, AGAP1. Okay? So we're just going to look at these guys and the GEFs here and how they're involved in recruiting the gigas and AP3. So the GEFs are a pretty large family. They have all a common conserved domain called the SEC7 domain. So this is the active domain of the GEF. So here's GBF1 and big 1 and big 2, which are rather large proteins. And there's a bunch of other GEFs that are involved in activating other ARF isotypes. Okay? But we're going to only be interested in GBF1 and big 1. Okay, so we're here at the Golgi. We want to recruit the gigas. You have a nice dileucine motif here in the tail that the VHS domain of the giga is going to bind. ARF1 GDP is going to be activated to GTP. And if you only want to recruit the gigas, is there a specific GEF that's going to be involved in this activation pathway? So, if you take cells and you stain them for big 1 and giga 3, here's big 1 staining in the Golgi, giga 3 staining in the Golgi, they kind of co-localize. If you use RNAi to deplete big 1, you still have giga 3 on the membrane. So although big 1 is going to activate ARF1, if you get rid of big 1, you're not affecting giga 3 recruitment. If you do the same thing with GBF1, so here you have GBF1, giga 3 in this case, there's a nice co-localization. You deplete GBF1, so here you have 3 cells, one cell that's pretty much completely depleted of GBF1, this one that still has a little bit, and this one that's still expressing quite a bit. When you look at the level of giga recruitment, you see that in the cells that are completely missing GBF1, you have very little recruited giga 3. And if you start expressing a little bit or a little bit more, you see a recruitment of giga 3 in these cells. If you look at AP1, which is also being recruited at the level of the Golgi, which is also being recruited by ARF1, GBF1 depletion has no effect on its recruitment. And here's a quantification of multiple cells. So, the GEF that's going to activate ARF1 specifically and only for giga recruitment is GBF1. So if we keep adding to our diagram here, here's the receptor, the gigas, ARF1. So now we can put GBF1 here in the diagram. That's going to activate ARF1 GDP to GTP. That's going to stabilize giga recruitment to the membrane, binding the receptor, and you'll form a nice trafficking vesicle. Okay? That's going to bring the cargo from the Golgi to the endosome. Questions? Total confusion?
What is it about how the different GEFs activate ARF1 that allows it to have specificity?
That's an excellent question. That's the next section. Okay, so if we look then, in this case, at the recruitment of AP3, so you have RF1 GDP to GTP. So can we find a GEF that's going to be involved in recruitment here? So you have AP3, and here's some AP3 staining. This is one cell, these are multiple cells. If you deplete, we can forget these two panels here, but if you deplete big 1, you see that you lose recruitment of AP3 in these cells, and you have a significantly diminished recruitment of AP3 in these punctate structures here. Okay? So AP3 is going to be recruited by big 1. So we can add big 1 to our diagram here. So RF1 GDP to GTP. So the recruitment of AP3 to move these integral membrane proteins from the Golgi to the lysosome is going to be recruited by big 1. Okay, so there's your question on how the GEFs are regulated. I think if you guys want, we can take a 5-minute break for you to get some shake out your legs, and we can come back and look at how the GEFs are regulated in 5 minutes. Okay? No? Everything's good? Okay. So, what have we looked at so far? You want to move a protein from the Golgi to the endosome. So there's two types of proteins that you're moving from the Golgi to the endosome. There's soluble proteins, cathepsins, prosapsin, those guys, and there's integral membrane proteins, LAMP and LIMP, for example. Okay? So, each one of them has to somehow interact with cytosolic trafficking components. So gigas, EAPs, to form clathrin vesicles to traffic. Okay? So the soluble ones are going to interact with a receptor. Okay? What are the two receptors? There's a mannose-6-phosphate receptor and sortilin. Okay? So they'll bind cargo through their luminal domain. Their cytosolic tails will bind what clathrin adapter to traffic from the Golgi to the endosome. So they're going to bind the gigas via their what motif? The dileucine motif. Okay? So now, the dileucine motif is going to bind the gigas. The gigas are going to bind clathrin. You're going to form a trafficking vesicle. What protein is regulating the binding of the gigas with the membrane? RF1. Okay? And in the case of the gigas, what protein is then regulating RF1 activation? GBF1. Okay? So all of those steps have to occur to make that trafficking vesicle. Now, in the case of LAMP2 or LIMP, every one of those players, or there's an equivalent of every single one of those players, the names just change. Okay? What makes it simpler for those integral membrane proteins is that they have their own cytosolic tail, so they're not binding a receptor. So they're going to bind directly to the cytosolic component to form their clathrin-coated vesicle. You still have RF1 recruiting that AP, so AP3. But this time, instead of GBF1 controlling that activation, what GEF is activating RF1 for AP3 recruitment? Was big 1. Okay? Bottom line is transmembrane protein, clathrin adapter, small g protein, GEF. Okay? All you gotta do now is, depending on which transmembrane protein, the names change for the different proteins. Okay? So you have a GEF regulating a small g protein, which is regulating the recruitment of a clathrin adapter, which is binding to a transmembrane protein via one of the motifs to form trafficking vesicles. Okay? So now, you had the question, how are the GEFs regulated? So you want the GEFs to go to the right membrane compartment to activate the appropriate small G protein to recruit the clathrin adapter. All right, and the way the GEFs know where to go are through this group of lipids called phosphatidylinositol. Okay? And basically, we're only going to look at two of them, so PI4P and PI3P. Okay? So now, why are we interested in these lipids? Okay? So, what we know is that these lipids are distributed in a unique fashion. Okay? And people call them sort of the signpost of an organelle. So each organelle tends to have a specific phosphatidylinositol that says, hello, I'm Golgi, hello, I'm endosome, or hello, I'm plasma membrane. Okay? And PI4P tends to be enriched in the Golgi. PI45P2 tends to be enriched at the plasma membrane. And PI3P tends to be enriched at the lysosome. So, we have enzymes that make these PI4Ps. Okay? They're not, the names of the enzymes aren't particularly interesting to you. What I do want you to remember is that PI4P tends to be enriched at the level of the Golgi. Okay? So the enzymes that make PI4P tend to hang out at the level of the Golgi to make PI4P for the Golgi. Now, if you use drugs to block the formation of these lipids, so LY over here will block PI3P synthesis, PAO will block PI4P synthesis, and Workmanin's kind of a dirty drug because depending on the concentration, it will block PI3P and or PI4P. But the bottom line is, if you look at GBF1, so here's the Golgi, giantin, so there's 3 cells here, a bunch of cells here. And now you look at GBF1 recruitment and GIGA3 recruitment and AP1 recruitment. They're all going to the Golgi. If you treat with a PI3P inhibitor, you're still recruiting GBF1. You're still recruiting GIGAs, and AP1 here is our control. If you use PAO, which blocks PI4P synthesis, and Workmanin in this case, which is also blocking PI4P synthesis, you see that GBF1 is no longer being recruited to the level of the Golgi. So now, if you're not recruiting GBF1 to the Golgi, what's happening? Part of the answer is on the slide. So what's happening here? So you're not recruiting the GIGAs if you're not recruiting GBF1. I told you that GBF1 was important for recruitment of the GIGAs because GBF1 does what? Right, so GBF1 is activating ARF1 specifically to stabilize GIGA recruitment to Golgi membrane. So if you don't have the GEF, you're not making ARF1, you're not recruiting AP. Okay? Well, if you look at AP1, it uses a completely different mechanism, and it still gets recruited to these Golgi membranes. So, if we can try to be a little bit more specific, we can try to identify which enzyme is making the PI4P for GIGA recruitment. And I'll make a long story short, that if you knock out this 3-alpha isoform, you completely lose GBF1 and GIGA3 recruitment. If you block this PI3 beta isoform, you don't. So even within the PI4P, you have control of exactly who you're being able to recruit. Okay? So the cell puts a lot of effort controlling the recruitment of these proteins and the formation of trafficking vesicles. Okay? For your point of view, PI4P is going to be specific to recruit GBF1 to activate ARF1 specifically for Giga recruitment. Okay? So this is basically what I said here. So here you have the enzyme that's making the PI4P. It's enabling GBF1 to be recruited to the membrane. GBF1 can then activate ARF1, GDP to GTP. You can stabilize Giga's interaction with the membrane, and the VHS domain can then bind to the receptor to form a trafficking vesicle. Okay? All right. So if PI4P was mostly here at the Golgi, PI3P is mostly here in these endosomes. And this time we're looking at retromer recruitment. So here are some cells stained with UPS26, one of the retromer subunits. You have nice punctate staining. SNCS1 is the same. It's part of retromer, SNCS2 as well. This time if you're treating with Wortmanin, so this was done at a concentration that you're only blocking PI3P synthesis. You see that you're blocking the recruitment of retromer, so either shown by UPS26 staining, SNCS1, or SNCS2. The protein is really staying in the cytosol and not being recruited. Okay? So in this case, you have retromer being recruited, you have PI3P being synthesized. The identity of the GEF responsible for retromer recruitment is not known, that's why it's been left off. But you need this PI3P synthesis and this drive of PI3P to recruit retromer to endosomal membranes. Okay? So PI4P at the level of the Golgi, PI3P at the level of the endosome. Any questions on this? All good? Okay. So now, we formed our vesicle. We've been able to regulate the formation in space and in time at different organelles to stabilize retromer on the membrane. Retromer can bind the receptor tail through wet motif, tyrosine motif, and you can form trafficking vesicles leaving the endosome and going back to the Golgi. Okay? So the basic premise is that you have the receptor that needs to move from point A to point B and point B back to point A. You have a clathrin adapter, either the gigas or retromer, that get recruited and bind specific membrane, specific domain within the tail of the receptor. Leucine, tyrosine. To control that interaction and to control trafficking, you have a small G protein. At the level of the Golgi, it's ARF1. At the level of the endosome, it's RAP7. Okay? So, just to show you that RAP7 is actually involved in recruitment of retromer. So if you have control cells or RAP7 depleted cells, and you look at BPS26 staining, you see this punctate endosomal staining, and if you deplete RAP7, you lose this endosomal recruitment of retromer, as shown here by this BPS26 staining. So AP3, we haven't really talked about it yet, coming up. So it's an adaptive protein at the level of the Golgi, maybe a little bit endosome. It's not affected by RAP7 depletion, and neither is AP1. Okay? So RAP7, specific for the recruitment of retromer. Okay. So now the last group of proteins, the multimeric adapters. So there's AP1, 2, 3, and 4. And they're all these multimeric adapter protein complexes. So they all have a large beta subunit. So beta 1, 2, 3, 4. They have a medium subunit called mu, 1, 2, 3, 4. And a small subunit called sigma, 1, 2, 3, 4. And then they become a bit more divergent with this other large subunit where you have gamma for AP1, alpha for AP2, delta for AP3, and epsilon for AP4. Okay? So, where do they work? So AP1, let's sort of disregard it for this context, but it is involved in endosome to Golgi trafficking. AP4 is going to sort proteins from the Golgi to the basolateral membrane of polarized cells. AP2 is going to be involved in endocytosis from the plasma membrane to the endosome. And AP3 functions in this pathway as well for another class of proteins which we're about to discuss. Okay? So just to sort of give you an idea, but I don't want you to focus on all of these adapters. Just keep in mind the gigas, retromer, and AP3 now that we're about to discuss. Okay? So what does AP3 do? So the protein content at the level of the lysosome has these soluble proteins like cathepsins that are involved in catabolism. But the lysosome also has integral membrane proteins. So for example, LAMPs, which are highly glycosylated proteins that are transmembrane proteins, that the majority of the protein is found in the lumen of the organelle, and they're highly glycosylated to form a special layer protecting the lysosome from the degradative enzymes inside. So the LAMPs sort of serve this function as a barrier preventing the cathepsins from chewing through the lysosomal membrane to escape outside the lysosome. Okay? So you have a bunch of other of these transmembrane domain proteins. So if it's a transmembrane domain protein, it doesn't need to bind the receptor to traffic. It's got its own machinery pre-built. Okay? You have the bulk of LAMP2 within the lumen of the membrane. You have a transmembrane domain. You have the carboxy terminal end of the protein, which is in the cytosol. Okay? And they have a couple of different signals. So they also have tyrosine signals, and they have dileucine motifs as well. Okay? So different proteins have different types of trafficking motifs. In this case, the cell wanted to confuse us all. Both of these motifs serve as Golgi to endosome trafficking. Okay? It's not clear why tyrosine motifs for the sorting receptors work in the other pathway, but in this case here, we can focus on these dileucine motifs. So the multimeric adapters combine both these dileucine motifs and these tyrosine motifs. Okay? And the tyrosine signals on, for example, AP3 will bind the mu subunit, and the dileucine signals will bind a pocket formed by delta and sigma 3. Okay? And if we go to our yeast 2 hybrid experiment, you can see here that these proteins that contain a dileucine motif, so LIM2, you need to express gamma and sigma for them to bind, while as LAMP2, which has a tyrosine motif, will bind straight to mu3. Okay? So this is shown here on this yeast 2 hybrid. Okay? So now, you have the membrane of the Golgi here, you have LAMP that needs to go to the lysosome, you have AP3 that's going to be recruited to the membrane, bind the motifs here, so the dileucine motif, and it'll bind plasma. So what happens if you don't have AP3 in the cell? So this is another one of these proteins that gets trafficked through AP3. So it goes to the lysosome, and if you look at the cell surface, there's very little of the CD63 on the cell surface. If you have AP3 deficient fibroblasts, CD63 doesn't get targeted directly from the Golgi to the endosome. Instead, it sort of wanders around the cell a bit aimlessly, and it ends up a lot on the cell surface. So if you do FACS analysis, you can see that in AP3 deficient cells, CD63, LAMP1, and LAMP2, so all these proteins are trafficked by AP3. In the deficient cells, you have a lot of it on the cell surface, while as the mannose-6-phosphate receptor, which uses what to traffic? That is mannose-6-phosphate or sortilin get from the... Yeah, but yes, but that's from endosome to Golgi, from Golgi to endosome. Yeah, but from the Golgi to the endosome, the receptors will bind the gigas to traffic. Okay, so you see that these guys don't end up on the cell surface when you lose AP3. Okay, so if If AP3 is coming onto the membrane, you obviously have a small ARF, small G protein, that's regulating this interaction at the level of the Golgi. And which small G protein is regulating recruitment at the level of the Golgi? ARF1? ARF1. Okay, you can see that AP3 can interact with ARF1. If you do a co-IP here or use 2 hybrid, you see that AP3 goes through this pull-down assay or through this U2 hybrid, can bind activated ARF1 very well, and that the binding, again, is through this sigma delta binding pocket. Okay, and again, here if we treat the cells with BFA, so this is this fungal metabolite that will block ARF1 activation. You can see AP3 here, staying nicely, level of the Golgi and in some puncta, AP1 as well. And if you add cells to BFA, you see that AP3 and AP1 end up in the cytosol, so they're not being recruited. So you lose the ability to recruit when you don't have active ARF1. Okay, so now, you should all be hopefully asking the question, so at the Golgi, you have ARF1, an activated ARF1, that's recruiting AP3 and GIGA. So now, if you're activating ARF1, and let's say you want to move LAMP from the Golgi to the endosome, you're going to need to recruit AP3. But you don't necessarily want to recruit GIGA, because you're moving LAMPs. But you're activating ARF1. So if you're activating ARF1 at the Golgi, shouldn't you be recruiting the GIGAs? So how can the cell regulate the activity of ARF1 to make sure you're only recruiting AP3 or GIGAs at the level of the Golgi? Okay? And that's through a group of proteins called the GEPs. Okay, so ARF1 in an inactive GDP form will be activated to GTP. Okay? This will allow it to bind the membrane and to recruit either AP3 or the GIGA. And the activity or activating ARF1 from GDP to GTP is done by a GAP, a guanine nucleotide exchange factor. Okay, and at the level of the Golgi, there's two gaps. There's big 1 and its homolog big 2, and there's GBF1. Now, you have this active ARF. You don't want ARF to be active all the time. I'm sure some of you work on cancers, and if you have these proteins that get turned on, and it's always on, what happens? You get cell growth, weird migration, all kinds of different things, so that's not good for the cell. So you want to shut off ARF1, and that's done through a GAP, a GTPase activating protein, and for example, AGAP1. Okay? So we're just going to look at these guys and the GAPs here and how they're involved in recruiting the GIGAs and AP3. So the GAPs are a pretty large family. They have all a common conserved domain called the SEC7 domain. So this is the active domain of the GAP. So here's GBF1 and big 1 and big 2, which are rather large proteins. And there's a bunch of other GAPs that are involved in activating other ARF isotypes. Okay? But we're going to only be interested in GBF1 and big 1. Okay. So we're here at the Golgi. We want to recruit the GIGAs. You have a nice isoleucine motif here in the tail that the VHS domain of the GIGA is going to bind. ARF1 GDP is going to be activated to GTP. And if you only want to recruit the GIGAs, is there a specific GEP that's going to be involved in this activation pathway? So if you take cells and you stain them for big 1 and GIGA3, here's big 1 staining in the Golgi, GIGA3 staining in the Golgi, we kind of localize. use RNAi to deplete big 1, you still have giga 3 on the membrane. So although big 1 is going to activate ARF1, if you get rid of big 1, you're not affecting giga 3 recruitment. You do the same thing with GBF1. So here you have GBF1, giga 3 in this case, with a nice localization. You deplete GBF1. So here you have 3 cells. One cell that's pretty much completely depleted of GBF1. This one that still has a little bit. And this one that's still expressing quite a bit. When you look at the level of giga recruitment, you see that in the cells that are completely missing GBF1, you have very little recruited giga 3. And if you start expressing a little bit or a little bit more, you see a recruitment of giga 3 in these cells. If you look at AP1, which is also being recruited at the level of the Golgi, which is also being recruited by ARF1, GBF1 depletion has no effect on its recruitment. And here's a quantification of multiple cells. So, the gap that's going to activate ARF1 specifically and only for giga recruitment is GBF1. So if we keep adding to our diagram here, here's the receptors, the gigas, ARF1. So now we can put GBF1 here in the diagram. That's going to activate ARF1 GDP to GTP. That's going to stabilize giga recruitment to the membrane, binding the receptor, and you'll form a nice trafficking vessel. Okay? That's going to bring the cargo from the Golgi to the endosome. Question? Total confusion?
What is it about how the different guests activate our one that allows it to have specificity?
That's an excellent question. That's the next section. Okay, so if we look then, in this case, at the recruitment of AP3, so you have ARF1 GDP to GTP. So can we find a gap that's going to be involved in recruitment here? So you have AP3, and here's some AP3 staining. This is one cell. These are multiple cells. If you deplete, we can forget these two panels here, but if you deplete big 1, you see that you lose recruitment of AP3 in these cells, and you have a significantly diminished recruitment of AP3 in these punctate structures here. Okay? To move these integral membrane proteins from the Golgi to the lysosome is going to be recruited by big 1. Okay, so there's your question on how the GEFs are regulated. I think if you guys want, we can take a 5-minute break for you to get some, shake out your legs, and we can come back and look at how the GEFs are regulated in 5 minutes. Okay? So, what have we looked at so far? You want to move a protein from the Golgi to the endosome. So there's 2 types of proteins that you're moving from the Golgi to the endosome. There's soluble proteins, cathepsins, prosapsin, those guys, and there's integral membrane proteins, LAMP and LIMP. Okay? So, each one of them has to somehow interact with cytosolic trafficking components. So gigas, APs, to form clathrin vesicles to traffic. Okay? So the soluble ones are going to interact with a receptor. Okay? What are the 2 receptors? The mannose-6-phosphate receptor and sortilin. Okay? So they'll bind cargo through their luminal domain. Their cytosolic tail will bind what clathrin adapter to traffic from the Golgi to the endosome. So they're going to bind the gigas via their what motif? The dileucine motif. Okay? So now, the dileucine motif is going to bind the gigas. The gigas are going to bind clathrin. You're going to form a trafficking vesicle. what protein is regulating the binding of the gigas with the membrane? R1. Okay? And in the case of the gigas, what protein is then regulating R1 activation? GBF1. Okay? So all of those steps have to occur to make that trafficking vesicle. Now in the case of LAMP2 or LIMP, every one of those players, or there's an equivalent of every single one of those players, the names just change. Okay? What makes it simpler for those integral membrane proteins is that they have their own cytosolic tail, so they're not binding a receptor. So they're going to bind directly to the cytosolic component to form their class-encoded vesicle. You still have ARF1 recruiting that AP, so AP3. But this time, instead of GBF1 controlling that activation, what GEF is activating ARF1 for AP3 recruitment? With big one. Okay? Bottom line is transmembrane protein, clathrin adapter, small g protein, GEF. Okay? All you got to do now is, depending on which transmembrane protein, the names change for the different proteins. Okay? So you have a GEF regulating a small g protein, which is regulating the recruitment of a clathrin adapter, which is binding to a transmembrane protein via one of the motifs to form trafficking vesicles. Okay? So now, you had the question, how are the GEFs regulated? So you want the GEFs to go to the right membrane compartment to activate the appropriate small g protein to recruit the clathrin adapter. All right, and the way the GEFs know where to go are through this group of lipids called phosphatidylinositol. Okay? And basically, we're only going to look at two of them. So PI4P and PI3P. Okay? So now, why are we interested in these lipids? Okay? So, what we know is that these lipids are distributed in a unique fashion. Okay? And people call them sort of the signpost of an organelle. So each organelle tends to have a specific phosphatidylinositol that says, hello, I'm Golgi, hello, I'm endosome, or hello, I'm plasma membrane. Okay? And PI4P tends to be enriched in the Golgi. PI4-5P2 tends to be enriched at the plasma membrane. And PI3P tends to be enriched at the level of the endosome. So, we have enzymes that make these PI4P. Okay? They're not, the names of the enzymes aren't particularly interesting to you. What I do want you to remember is that PI4P tends to be enriched at the level of the Golgi. Okay? So the enzymes that make PI4P tend to hang out at the level of the Golgi to make PI4P for the Golgi. Now, if you use drugs to block the formation of these lipids, so LY over here will block PI3P synthesis, PAO will block PI4P synthesis, and workmannin is kind of a dirty drug because depending on the concentration, it'll block PI3P and or PI4P. But the bottom line is, if you look at GBF1, so here's the Golgi, giantin, so there's 3 cells here, a bunch of cells here. And now you look at GBF1 recruitment and GIGA3 recruitment and AP1 recruitment. They're all going to the Golgi. If you treat with a PI3P inhibitor, you're still recruiting GBF1. You're still recruiting GIGAs, and AP1 here is our control. If you use PAO, which blocks PI4P synthesis, and workmannin in this case, which is also blocking PI4P synthesis, you see that GBF1 is no longer being recruited to the level of the Golgi. So now, if you're not recruiting GBF1 to the Golgi, what's happening? Part of the answer is on the slide. So what's happening here? So you're not recruiting the gigas if you're not recruiting GBF1. I told you that GBF1 was important for recruitment of the gigas because GBF1 does what?
Yes.
Right, so GBF1 is activating ARF1 specifically to stabilize giga recruitment to Golgi membrane. So if you don't have the gap, you're not making ARF1, you're not recruiting AP, the giga, okay? While if you look at AP1, it uses a completely different mechanism, and it still gets recruited to these Golgi membranes. So, if we can try to be a little bit more specific, we can try to identify which enzyme is making the PI4P for giga recruitment. And I'll make a long story short, that if you knock out this 3-alpha isoform, you completely lose GBF1 and giga-3 recruitment. If you block this PI3 beta isoform, you don't. So even within the PI4P, you have control of exactly who you're being able to recruit, okay? So the cell puts a lot of effort controlling the recruitment of these proteins in the formation of trafficking vesicles, okay? For your point of view, PI4P is going to be specific to recruit GBF1, to activate ARF1, specifically for giga recruitment, okay? So this is basically what I said here. So here you have the enzyme that's making the PI4P. It's enabling GBF1 to be recruited to the membrane. GBF1 can then activate ARF1 GDP to GTP. You can stabilize giga's interaction with the membrane, and the VHS domain can then bind to the receptor to form a trafficking vessel. Okay? All right. So if PI4P was mostly here at the Golgi, PI3P is mostly here in these endosomes. And this time we're looking at retromer recruitment. So here are some cells stained with UPS26, one of the retromer subunits. You have nice punctate staining. SNX1 is the same part of retromer, SNX2 as well. This time if you're treating with Wortmannin, so this was done at a concentration that you're only blocking PI3P synthesis. You see that you're blocking the recruitment of retromer, so either shown by UPS26 staining, SNX1, or SNX2. The protein is really saying in the cytosol not being recruited. Okay? So in this case, you have retromer being recruited, you have PI3P being synthesized. The identity of the gap responsible for retromer recruitment is not known, that's why it's been left off. But you need this PI3P synthesis and this drive of PI3P to recruit retromer to endosomal membranes. Okay? So PI4P at the level of the Golgi, PI3P at the level of the endosome. Any questions on this? All good? Okay. So now, we formed our vesicle. We've been able to regulate the formation in space and in time at different organelles using all the levels of regulation that we've discussed. Okay? Now, we've made the vesicle, Dynamin cut the vesicle, and the vesicle is off and running to its target destination. How does the vesicle get there? So, we're going to add just a little bit of complexity here. So, this is a polarized cell of an epithelium, and you have an apical membrane and a basolateral membrane. And you have these aquaporins, so these water channels, that are being sorted from the Golgi in these clathrin-coated vesicles. And aquaporins go to very specific places. So you'll only find aquaporin 2 at the apical membrane, and you only find aquaporin 3 and 4 at the basolateral membrane. But they're both being, all three are being packaged into clathrin-coated vesicles. So how do the vesicles know to go specifically there or specifically there? And the bigger question, or another question you can ask is, how do they get there? So is it like you plug in your GPS, you want to go from IRCM, and I want to go back to work in Laval, and the GPS is going to tell me this is the shortest, fastest way of getting there. So if I go from point A to point B, I get back to work. Or do I leave my GPS at home and randomly drive to every university in Montreal until I find the university that I belong to? So we used aquaporins as a model for this because we knew they were packaged into trafficking clathrin-coated vesicles, and they localized to very specific places. Okay? So aquaporins are these multi-spanning membrane proteins. They know that the APs get recruited, you have an ARC that's controlling this recruitment, and you have the formation of trafficking vesicles. Okay. So the other thing we know is that vesicles tend to move on microtubules. Okay? So there's motors that move the vesicles along the microtubule. But is this sort of a direct highway? So this is the donor membrane and the acceptor membrane, the plasma membrane. Or how do they get to snares, like I told you? The pairing snares match up to where they have to go. Those are the snare set. So the bottom line is, do they have direct transport? Do they bud from the Golgi, traffic on these microtubules, and go straight to their known destination and fuse? So that's the question we want to answer. Okay, so how can we answer these questions? So we know that aquaporins go to a very specific membrane domain. So if you have two cells, and you're looking at aquaporin 3 over time, you can see that the cells, so these points of contact, the cell thinks it's polarizing, so they're touching one another. So I think it's a basolateral membrane. So it's trafficking aquaporin 3 to that site. And over time, you can see aquaporin 3 accumulate. You can take single confocal images of aquaporin 4, and you can see that it's in the basolateral membrane. And these techniques are sort of nice, so you can do RNAi, you can delete things, try to make aquaporin 4 to move to different places, but you're neglecting any dynamics of the trafficking. So another thing you can do is you can do some live cell imaging, and you can track vesicles over time. And these guys followed one vesicle, and they made a nice trajectory of where the vesicle was going. But that was one vesicle. So is that one vesicle representative of every trafficking vesicle for, say, aquaporin 4? So what did we do? So we transfected GFP aquaporin 4 into cells, and we could do some live cell imaging. So these are the aquaporin 4 containing vesicles. And you can see that they're moving. So if you want to do image analysis on these vesicles, the computer doesn't like all of the background. So if you can filter out some of the background light, you're left with just the trafficking vesicles, as you can see here. And then you can write a computer program that can detect tracking of these vesicles, try to figure out where they're going, how fast they're going, and a bunch of other things. So if you put all of the vesicles together, you can see, you can follow hundreds and thousands of vesicles by doing this image analysis to get an idea of, travel, how far they went, which way they went, how fast they went, different parameters. Okay? So the data I'm going to show you is only on basolateral membrane trafficking. It's easier to follow the vesicles moving this way than this way on the plane, because the vesicles there change plane. So you have to do a 3D analysis, which is beyond our capabilities of writing computer programs right now. Hopefully we'll get better. But we can follow them going this way, so it's a bit easier. So bottom line is, we ask a rather simple question in a kind of complicated mathematical formula. So basically, every one of the tracks gets an ellipse fitted around the track. And if you do all the fancy math and a bunch of other things, you end up with two numbers, 0 or 1. Okay? Basically, 0 means the vesicle was doing this, so it wasn't going anywhere, and 1, it moved from point A to point B in a perfectly straight line. Okay, so we tested our ability to track these guys. So these are fluorescent particles on a cover slip, and if you do nothing to the cover slip, the fluorescent molecules just diffuse randomly, and we can follow their tracks. But if you move the stage in a direction, all the particles sort of follow the direction of the stage. And you can see that their path lengths are much more elongated, or closer to 1. So if you do an analysis of these guys, and you plot it, so the red line is this random motion, and you can see that most of the vesicle paths in this random motion are at 0 or tend to be closer to 0. And with the motor moving, where you have these nice straight lines, most of the paths are moving towards a value of 1, so in a straight line. Okay? So our algorithm could identify the vesicles and track them, and our A2 value of 0 or 1 is represented here. So our algorithm is working, so our system's set, ready to go. So can we learn anything about how these vesicles move now? Okay, so the theory we had is that if you look at aquaporin 4 trafficking in unpolarized cells, so there's no basolateral membrane. So aquaporin 4 doesn't have anywhere to go, right? So, it's supposed to go to the basolateral membrane. There is no basolateral membrane. It's going nowhere. So it should be basically just randomly wandering around. If you look at a polarized cell, so these two cells touching here, and you're looking at aquaporin 4 as this basolateral membrane, we figured, well, okay, aquaporin 4 has a place to go. It's going to leave the Golgi and go to the basolateral membrane. All right? So could we see this? So here are these cells that we transfected. So these are the polarized cells, and you can see this basolateral membrane. So we looked at 150 cells in this condition, and we quantified 20,000 vesicles. And in the non-polarized cells, we did 50 cells and 27,000 vesicles. So you can see here in the polarized cells, the aquaporin 4 is really going towards this basolateral membrane, and it's accumulating there. While as in the non-polarized cells, it's hanging out in these vesicles because it's got nowhere to go. All right? So, if you track these vesicles, so these 20,000 vesicles here and these 7,000 vesicles here in these two conditions, what do we see? We see absolutely nothing. So the vesicle dynamics are actually the same in the non-polarized cells and the polarized cells, which frankly was a big shock to us because everybody in the field assumes that the cell's going to want to spend the least amount of possible energy moving these vesicles around, so the cell's going to want to move the vesicles from point A to point B in the shortest amount of time and the shortest distance possible. So we all assume that the vesicles would go straight to their destination. But that doesn't appear to be the case. And we looked at a bunch of different descriptors, so the path length, so how far the vesicles move, the direction, the velocity. Both cases, it was identically the same. All right? So that was kind of disappointing. So then we thought, well, okay, maybe the vesicles sort of leave the Golgi, wander around a little bit, and then when they get close to the basolateral membrane, they sort of, okay, this is where I have to go. Maybe they'll start heading off in a straight direction then. So we looked at vesicle movement close to the basolateral membrane, so 10,000 vesicles in each condition, and no difference. So many thought, well, okay, maybe aquaporin 4 is just stupid. Maybe it doesn't know where it's going, so let's try aquaporin 3. We did the same thing again with aquaporin 3, and it's identical data to aquaporin 4. So that's kind of weird. So then we thought, well, okay, maybe the vesicles are not really being trafficked for some reason. Maybe it's because of the GFP tag, or maybe we're just studying some sort of an artifact, our data. And in fact, that's not what we're seeing. So the vesicles are not really randomly just diffusing. They're actually going somewhere. But they're not actually going from point A to point B. At least not the way we assumed or thought that they would go. So if they were being moved mechanistically, so there was a motor moving them, was a microtubule that was moving it. So we depolymerized microtubules using a drug. We also did it for actin. Now, I'm a biologist. When my physics postdoc showed me these images, I was very excited because we could clearly see depolarized microtubules, but being physicists, they don't believe anything with their eyes. They need a computer to prove that they're depolarizing the microtubules. So my postdoc wrote another algorithm to determine the smoothness of these microtubules to see if we could actually quantify the depolarization. And we segmented the imaging, and lo and behold, what our eyes were telling us, the computer was telling us, and my physics colleagues were happy with the data. So yes, we were depolarizing microtubules and actin as well. So what happened to vesicle dynamics in this case? So here's a cell expressing GFP aquaporin 4. You can see that the vesicles are moving around quite a bit. After 3 hours of treatment to depolymerize microtubules, the vesicles really started to slow down and move in a more random motion. So the vesicles were being targeted, they were being moved mechanistically through microtubules, but they weren't quite going directly from point A to point B. So that was kind of surprising. So, we know that they're moving through the microtubules, but they're not moving in sort of quite the fashion point A to point B. Can anybody think of a hypothesis or what might we be missing? So the study we did was in non-polarized cells and polarized cells. So what could be happening in those situations? And is there a situation where theoretically or maybe we could see that the vesicles were being targeted more directly to their basolateral membrane?
Attractant? Maybe an attractant?
Maybe it's an attractant. So what we're trying to do now is, based on the data we got, we thought.
Sorry, I have a question. Usually vessels transport some protein. This protein inside the vessel doesn't play a role in dictating the target inside. Because some proteins, like some post-translation modifications, they are not, even they are in the vessel, they are not well transported.
Well, that's possible, but aquaporin 4 in this case is getting, so it's going through the Golgi, so it's in theory getting any post-translational modifications that it's supposed to be getting. And we're looking at a post-Golgi event. So that, unless the GFP tag is somehow interfering with something, that's possible. But what we're trying to do, and so what we're looking at sort of, or we think that we're looking at two extreme states where the cells are completely non-polarized and completely fully polarized. So when they're fully polarized, they have aquaporin 4 on the basolateral membrane. So they're probably trafficking some amount of aquaporin 4 to maintain expression levels, and they're probably internalizing some to get degraded. So we're wondering if we can try to catch cells during polarization when there's a need to set up that aquaporin 4 on the basolateral membrane if we'll see more directed trafficking. So we'll see. So at least for these kinds of vesicles, they don't really go from point A to point B. So what they seem to be doing is they hitch a ride on microtubule and they go someplace. But vesicles walk on these microtubules with dynein arms. And what could be happening, and the other thing we're trying to test is, maybe it's completely snare dependent, where the vesicles will go, they'll sample membranes, and if they don't have the correct snare pair, they won't fuse there, they'll hitch a ride back on the microtubule and go someplace else until they find that target membrane. So those are the two possibilities. Okay. Questions? Okay. So, the last part. So, I really get excited about vesicular trafficking. I'm hoping at least some of you are getting somewhat excited about vesicular trafficking. But a good way to sort of get excited about it is, of course, most of the proteins any of us work on, is somehow related to disease. So are there any vesicular trafficking associated diseases? So one of them is from a mutation on this epithelium sodium channel. So it's a 3-protein channel that's expressed especially in the kidney, lung, and sweat glands, and it essentially regulates water and sodium levels. Okay? So if any of you do any work on the kidney, what is the kidney doing with water and sodium levels? What's that regulating? Blood pressure. Okay? So these guys are really important in regulating blood pressure. So these guys are in a polarized cell. So these sodium pumps are sitting at the apical membrane of the kidney cell, and they're internalizing sodium and water. Okay? So the cell can put more or take away receptors through this pathway. So in this case, AP2. So we didn't really talk about it, but AP2 will form placental vesicles here at the cell surface, and it'll bring in proteins to be degraded. Okay? So this sodium channel is sitting here on the cell surface, and depending on how the kidney wants to regulate blood pressure, it'll put more or less of this transporter on the cell surface. So, the ENaC, the beta subunit of the sodium transporter, has a tyrosine motif in this case, which is known to bind to AP2 and form these trafficking vesicles so that the cell can remove these transporters from the cell surface. So some patients, and it's a dominant genetic disease, have a mutation in this tyrosine motif where the tyrosine is replaced with a histidine. Okay? So what that results in is inefficient endocytosis of this protein. So what happens in the kidney is that you have a salt and water balance that the cell can't regulate. So the patient ends up with this ridiculous hypertension at very early ages. Okay? So the patients are treated pretty simply. They don't eat too much salt and a couple of other things, and they seem to generally be okay. But you get this gain of function mutation where the cell can't regulate the trafficking of this transporter, and the patients get hypertension. Okay? And some of the other diseases involved in trafficking are these lysosomal storage disorders. So they're fairly rare group of over 60 different diseases. They have about 1 in 13,000 live births. The major issue with these kids is neurological dysfunction. You can also have organomegaly. So this young man here has a very distended abdomen because he has a mutation in a lysosomal protein that's involved in degrading lipids. And so his liver can't degrade those lipids. So the liver accumulates those lipids, and the liver enlarges, and it distends the abdomen. Okay? They also have skeletal deformities and coarse facial features, as you can see in this young man here. So what's sort of really devastating of these diseases for the parents of these individuals is that these kids are born, and they start hitting normal life milestones. So they start growing properly, they develop, they can start walking, they can start talking, they have sort of normal development and interactions with their peers. Over time, substrates start accumulating so that the disease phenotype starts to progress. So kids, for example, who were able to walk, sometimes lose the ability to walk. Kids who were able to talk lose the ability to talk. There's also a high component of blindness with these diseases, so it's very difficult for the patient, for the parents of these patients, because you seem to have a normal kid, and then after 2 or 3 years, or depending on the disease, the kids really start to show really bad phenotypes, and death usually occurs for these individuals probably in the second decade of life. Okay? So, at a biological level, this is a slice of brain from one of these patients, and you can see here this yellow accumulating material called lipofuscin, So that's one of the hallmarks of a lot of these diseases. So this isn't a confocal image, this is just a regular immunofluorescence image where these guys, this lipofuscin autofluoresces yellow. And at an ultrastructural level, if you look at this multivesicular body and lysosome, these are sort of normal-looking multivesicular bodies and lysosomes, which have this electron-dense material. But if you have a mutation in one of these proteins that are degrading lipids, you can see that the multivesicular bodies, which should look like this, start looking like this, where they accumulate this high levels of weird lipid profiles. Okay, and eventually this causes the neurological defects and eventually death in these patients. Okay, so my lab studies this family of proteins. It's the CLN family, so there's 14 different genes that have been identified, and each one of them is one of these 60 lysosomal storage disorders or lysosomal diseases. We're particularly interested in CLN3 and CLN5. But the question was, you have CLN5. We knew that it was mutated in this NCL, so neuronal ceroid lipofuscinosis. It's one of these lysosomal diseases. But the function of the protein wasn't known. We knew that it was localized to the endosomes and lysosomes, but nobody knew what the protein was doing. Okay? What we did know is that it did interact with another family member, CLN3, and for some reason, well, there's a founder effect in Newfoundland where these patients with CLN5 mutations, there's 1 in 11,000 live births in Newfoundland, which is fairly high incidence of this disease, because the rest of the world you're looking at about 1 in 100,000. So there's a founder, some individuals in Newfoundland that spread a mutation in the CLN5 protein. So, what's CLN5 doing? Well, it's a soluble protein. So at first we thought, well, maybe let's just look at the way it travels. All right, so we did some co-IPs with sortilin and CLN5. Now, here's the trick here. So we did the IP at pH 5 and at pH 7. What did we find? So if you IPHA CLN5 and you stain for MYC, you find an interaction at both pH 5 and pH 7. Why is that kind of weird? So what do we talk about pH and binding to cargo receptors? Right, so the cargo receptor interaction should occur at a more neutral pH. And if you look at prosapsin, which is another sortilin binder, you see that prosapsin binds at pH 7, but doesn't bind at pH 5. But CLN5 seems to bind sortilin at a pH of 5, equally well as at a pH of 7. And if you deplete sortilin, you still have normal trafficking of CLN5, where the known sortilin cargo prosapsin, you had no more trafficking. So CLN5 was binding not really like a cargo, and if you deplete the receptor, the protein was still getting to its appropriate intracellular location. So we tried the inverse. So here we depleted CLN5, so we stained for giantin and the mannose-6-phosphate receptor and sortilin. Here you have the mannose-6-phosphate receptor that's localizing in the Golgi, which is where it should, and sortilin in the MOC-depleted cell. But if you deplete CLN5, you start seeing the receptors in these punctate structures. So now, if the receptor is supposed to be in the Golgi, and now you're seeing it in these puncta, so in these structures that remind you of endosomes, or they should, what's CLN5 doing to the receptors? So, what did we talk about? If you can't move the protein back from the endosome back to the Golgi, what happens to the receptor? It gets degraded. So, we did a cyclohexamide chase, MOC, CLN5 depleted cells, and CLN1, which is another one of these related proteins. And we looked at the stability of the protein, of the receptors, chromatocyte phosphate and sortilin with a mixed tag. The MOX depleted cells, the receptors are nice and stable. CLN1 depleted cells, the receptors are nice and stable. But in the CLN5 depleted cells, we see the receptors are degraded by 6 hours. And if you look here, this is an assay where we can try to determine the function of the lysosomal protein. So the lysosomal proteins get cleaved to mature form once they reach the lysosome. And if they have a problem reaching the lysosome, you see an accumulation of this intermediate form, which is what we're seeing here in the CLN5 depleted cells. So known cargos are having a hard time reaching it, presumably because the receptors are being degraded. Okay, so now, the receptors are getting stuck at the endosome, and if you look over time, they're getting degraded when you knock out CLN5. So in what condition did we see the receptors stay stuck at the level of the endosome and the receptor get degraded? Retromer. So, is CLN5 somehow involved in retromer function? Okay, so the receptors are getting stuck here, and they're going to get degraded. So maybe there's a problem with retromer. So we looked at retromer recruitment. So here are these MOX depleted cells, CLN5, retromer, staining with this UPS26. It gets recruited to these endosomal structures. You deplete CLN5, you see no more recruitment of retromer. If you deplete CLN1, which has been involved in the pathway, you see nice normal recruitment of retromer. Now, you could be skeptical of this data. Maybe you say, yeah, but we just found the 2 cells that prove your point. So we quantified the data. So again, we wrote a computer program that can identify the cells, then inside the cells finds all the punctate structures and quantifies the fluorescence intensity of each of the punctate structures. And if you analyze thousands of these endosomes, you can see in the MOX situation, if you set it at 100, when you deplete cells of CLN5, you have a 50% reduction in the recruitment of retromer to endosomal membrane. While if you deplete CLN1, you have no effect. Okay? The other thing you could argue is, well, maybe the retromer is being degraded in CLN5 depleted cells, but if you look at the total amount of retromer in these lysates, there's no change. So it's really a case where you're not recruiting the retromer to these endosomal membranes. And if you look, I told you before that RAB7 was involved in retromer recruitment, and I'm arguing now that CLN5 is, and you look at the distribution of retromer within the endosomes compared to the cytosol, you can see here in the MOX, you have about almost 60% of it on the membrane. If you deplete RAB7, you significantly decrease the amount on the membrane, and you see more in the cytosol, and you see the same thing when you deplete CLN5. Okay, so when you deplete CLN5, you're not recruiting retromer. I just told you what's important to recruit retromer. It's RAB7. What could be happening to RAB7 if you're not recruiting retromer? So what does RAB7 do? What is RAB7? Small g protein that gets activated from GDP to GTP. So what's happening to that? So again, if you look just by immunofluorescence, you deplete CLN5, you don't see any recruitment of RAB7, and we did the same quantification here. If you look at RAB5, which is another endosomal RAB that I didn't talk about, but you see that depleting CLN5 really doesn't have much of an effect on the recruitment of RAB5 to the endosome, when it has more significant effect on RAB7. The expression of both of these guys are normal, so they're not being degraded. So RAB7 isn't being recruited to endosomal membrane. So can we see if it's activated? So just to remind you, RAB7, GDP to GTP, depth and gap to control the cycle. And what we did here is we did a GTP loading assay. So we fed cells with this analog of GTP that's conjugated to a biotin, which under UV you can cross-link into a small G protein. So depending on the activity status of your favorite G protein, There'll be more of the probe or less of the probe, depending on what you're doing to your cell. And if you look here at the quantification of these plots, you can see that the amount of loaded GTP in the MOC versus the CLN5 depleted cells, you have a 70% reduction in GTP loading of RAB7 in CLN5 depleted cells. Okay? This is another assay to test for RAB7 loading. So GSP RILP is another protein that RAB7 interacts with, but RAB7 will only interact with RILP when it's activated. So if you do a GSP pull down with RILP, and you look to see how much RAB7 comes down, you see in the MOC and in the CLN1 depleted cells, you have a fair bit of RAB7 coming down. And when you deplete CLN5, you have significantly less RAB7 coming down. So you're not activating RAB7 when you're depleting CLN5. Okay, so we're here at the level of the endosome. Here's the receptor. Cargos come off. You need to recruit retromer. You need to activate RAB7. Somehow, when you deplete CLN5, you're not activating RAB7 and you're not recruiting retromer. Okay? What's the problem? So that could be, but CLN5 is here. There's a membrane here separating RAB7 and retromer. So a couple of months ago, a nice review on MCLs came out, and the writer of the review basically said, how can a soluble protein activate RAB7 to recruit retromer? This data is impossible. Well, I'll argue that maybe that's a bit small-minded, because I told you that RAB7, CLN5 also binds CLN3. Okay? So although CLN5 is inside the lumen, it's binding to a transmembrane protein CLN3, which is another founding member. Okay? So the other thing that happens is that CLN3 is known to bind RAB7. Okay? So possibly now you have a system through a transmembrane protein to activate RAB7. Okay. So how did we test this hypothesis? So we used BRET, bioluminescence resonance energy transfer. Anybody use this in your daily work? Anybody know what it is? You're using BRET? Huh? Oh, okay. So bottom line is, BRET, you can study protein-protein interactions in live cells and in real time. Okay? So you're not necessarily getting a snapshot of when you're doing co-IP, but you can do it in live cells. So what you do is you take one protein, you tag it with a luciferase, you take another protein with an acceptor, and if these two proteins interact, you add a substrate, the luciferase converts the substrate to a light source, and it can activate the acceptor. Okay? And depending on how you do your studies, you can do a nice curve. So if you see a saturation curve like this, it means that your two proteins interacted. If you see a straight line like this, it means there was no interaction. Okay? The nice thing with BRET is it's live cells, so you can do all kinds of different things with it. You can also test multiple interaction partners with the complementation assay. But you can also look at the effect of agonists on, say, GPCRs. So let's say this interaction only works when you stimulate the G protein coupled receptor with an agonist. You can see that in native state, there's no interaction. You add the agonist, then you see an interaction. So you can play around with these interactions and try to block them in live cells and in real time to really study protein-protein interactions and the dynamics of protein-protein interactions. Okay, so I told you that CLN3 binds RAB7. So can we see this interaction using BRET? And we have a nice curve. So yes, we can see an interaction of CLN3 with RAP7. Now, what does CLN5 do to these interactions? Okay, so there's a few too many plots here, but just look at the black bar and the red bar. So here we're looking at RAP7 interacting with CLN3. So here's the interaction. If you add CLN5, you're decreasing the interaction of CLN3 with RAP7. If you knock out CLN5, and we use CRISPR to knock it out, you see more interaction of CLN3 with RAP7. So somehow CLN3 is modulating that interaction. So whatever effect CLN5 is having on RAP7 and retromer recruitment, it's somehow mediating it via CLN3. So we're in the process of trying to figure this out. The model we have is, if you look at this data here, where if you lose CLN3, you see more of a RAP7 interaction. So what we think happens is the receptor arrives at the level of the endosome. The change in pH causes a release of the cargo. The receptor now is free to bind CLN5, so it pulls CLN5 off of CLN3. So it initiates that, it modulates the interaction of RAP7 with CLN3, and somehow that's involved in the activation and recruitment of retromer. Okay? So what we're trying to also determine is these are different mutations in CLN5 that are known to cause human disease. So we're trying to see if any of these mutations are affecting this CLN3 RAP7 interaction. Okay? So this experiment's kind of nice, but it was done just by overexpressing these guys with native CLN5 still present, so we're not quite sure about the data. But we're hoping to take advantage of these CRISPR clones now, where we eliminated endogenous CLN5, and we can put back in the different mutants and see what happens. We can try to figure out what's happening in these individuals who have CLN5 disease. Okay? Any questions? Okay. So I've told you. Yeah.
What if you know of
Okay, we haven't done it yet. So that's definitely something that's along the line. So CLN3, there's also a bunch of mutations in patients that have a similar disease to the CLN5 mutation. So the proteins are really closely related. Biology, but there's 3 patients around the planet, so, maybe it's not that big a deal. But let's talk about Alzheimer's disease. Okay, I'm sure everybody knows what Alzheimer's disease is. Neurodegenerative disease, loss of brain function, death, significance of Alzheimer's increases with age. And one of the big health problems we have is, especially in the Western world, people are living longer. And I think the last statistics I saw is 1 in 3 people over the age of 85 have some form of Alzheimer's disease. So it's clearly a significant health problem. It costs a lot of money to care for these people. It's difficult on the family members, because the patients are still sort of able-bodied, but have very diminished cognitive function. So what's the theory for Alzheimer's? So one of the theories is it involves this enzyme, beta site ATP cleaving enzyme called BACE. So it's a transmembrane protein with relatively unknown biological functions, although that's not completely true now. More and more, a couple of papers have come out to sort of show what BACE might be doing endogenously. So what's interesting about BACE? Well, it cleaves this protein called APP. Okay? And the bad cleavage goes this way. So if you make A beta peptide, the A beta peptide is secreted out of the neurons, it forms these plaques, and this is one of the theories around Alzheimer's. If you cleave the other way, you generate this P3 fragment, and this isn't a problem. So the beta secretase, so BACE is here on this pathway, while the alpha secretase is here on this pathway. So one of the things that people are trying to do and thinking about is you want to block this function to highlight this function. So what's one way of blocking this function? No? So how do you block or how do most drugs work? So BASE is an enzyme. It's cleaving this APP into this A beta fragment. So if you find a small molecule that you can just block the catalytic activity of BASE, in theory you have a medication to treat Alzheimer's disease. Okay? So, I mean, I'm not that smart. Other people have obviously thought about this before. So pharmaceutical companies have spent a lot of money trying to find inhibitors of BASE. They don't work. Okay? One of the thoughts is the catalytic pocket for BASE is kind of big compared to most cleaving enzymes. So fitting a small molecule in there to block its activity is really not working. Okay? So, but what about BASE then? So BASE is a type 1 transmembrane domain protein that traffics inside the cell. So it goes from the Golgi, goes to the endosome, and comes back. And most of the cleavage events occur here in the endosome. So BASE is a cleaving enzyme. It likes acidic environments. So it's going to cleave proteins in more acidic environments, like the endosome. Sortilin is elevated in Alzheimer's disease. So why is that important? Well, sortilin seems to regulate the processing of APP. So if you overexpress sortilin, you have more APP being A beta being produced. And if you degrade sortilin using shRNA, you have less A beta being produced. And if you look at the stability of BASE, it's directly related to the expression levels of sortilin. So if you look at the stability of BASE and sortilin over time, here's BASE in normal cells. If you deplete sortilin, you'll lose BASE from the cell. If you block RAB7, what's RAB7 going to do? It's going to block the activity, the cycling of the protein, and so BASE starts accumulating in these cells. So is there a way to somehow modulate sortilin trafficking to somehow affect BASE trafficking and decrease A beta peptide production? Okay, so we're here at the level of the endosome, and we want to degrade sortilin to maybe get rid of BASE. So if there's too much sortilin, it keeps BASE cycling, keeps BASE active, and you have more A beta peptide. So how could we treat these patients, theoretically, to cause that excess sortilin or to decrease the level of sortilin, which would then lead to decrease in BASE and hopefully less A beta peptide? So how do we do it? You guys know now. You should know. So what's the simplest way that we know to get sortilin instead of going this way to go this way? What pathway should we inhibit? So if we block retromer function, we should be able to send sortilin this way instead of this way, and in theory you should drop the levels of BASE. So how can we block sortilin function? Nope. That's sort of the same protein as sortilin in a way. So we could maybe somehow inhibit RAB7. We could block PI3P production, which would prevent RAB7 and retromer from being recruited. So there's other ways as well. So this is just to sort of highlight that this sort of fundamental pathway is involved in a lot of diseases, and this fundamental pathway is also involved in a lot of things. You don't really have immunology if this pathway doesn't function. For example, viruses also use this pathway to infect cells, so HIV, for example. So modulating this pathway, Leishmania infection, which is a parasite, which is not really a problem here in North America, but around the world is a significant health problem, uses this pathway to replicate itself and to cause infection. So understanding how this pathway is modulated and regulated could be a potential way of treating not only these rare genetic diseases, like these like. So this is where I work, the building where I work. This is me and my information. If you have comments, questions, If you're freaking out, don't hesitate to call me. Be happy to, or email me, be happy to answer questions, and I can hang around this a few minutes if anybody has